Several chapters of The Origin of Species concern themselves with Darwin's interpretation of the causes of the patterns of variation seen in domesticated and natural populations, as well as the effects of hybridism. Darwin's understanding of genetics lagged behind his knowledge of natural history of wild populations and even the results of his own home experimentizing, however. At the time of the publication of The Origin, Gregor Mendel had been working in his garden at the monastery at Brno and had a much better understanding of these ideas. Reading Mendel's major work from 1866 and seeing the annotations he made in his own copy of The Origin, it is clear that Mendel knew about Darwin's ideas and saw the applicability of his genetic work to it, but the connection was never really made between the two scientists. Transmission genetics is how genes are passed from parents to offspring. Mendel inferred the parental contributions to their offspring from crossbreeding experiments. Today we understand that two copies of a gene are located at a particular spot on physical chromosomes inside cells. His law of segregation tells us that only one copy of each gene at a particular locus is transmitted by the gametes to the offspring. He recognized that some alleles were dominant while others were recessive. In today's terms, we would say that the phenotype of recessive proteins can be masked by the phenotype of more dominant ones. For example, faster enzymes mask the catalytic rate of slower ones, or darker pigment proteins mask lighter pigments. The law of independent assortment is understood today to mean that genes that are on separate chromosomes, or sufficiently far away from one another on a single chromosome, are passed down independently of one another. So traits don't occur together unless their genes are linked, located physically close on a chromosome. Darwin had some sense of a blending style of inheritance that wasn't really borne out. The closest that we have are genes with incomplete dominance. The difference is that fully dominant or recessive phenotypes can be recovered in subsequent generations. If it were only about blending, new phenotypes would be mixed into oblivion after a few generations. On the other hand, before we drag Darwin too much for not knowing about genes, let's not forget that it took until 1928, 44 years after Mendel's death, for biologists to confirm that it was cellular DNA that was the genetic material. By the turn of the 20th century, and certainly in the post-war period, science as a profession had expanded to the point where we stopped talking about individual scientists like they were prophets. We hold up Copernicus for astronomy, or Vesalius for anatomy, or Lyell for geology. But from about the middle of the 20th century, we started talking more about experiments and papers, like Watson and Crick for the structure of DNA, or Messelson and Stahl for semi-conservative DNA replication, or MacArthur and Wilson for island biogeography. And at that, we have even looked at those people and started identifying and celebrating the work of people like Rosalind Franklin, scientists whose ideas were unacknowledged during their time. So, a brief rundown about what we understand about genetics now. For decades, we've known about what we call the Central Dogma, the idea that genes are segments of DNA that hold the information to build a given polypeptide. DNA is transcribed by RNA, and that messenger RNA is delivered to the protein-building area of the cell. Once there, ribosomes translate that mRNA sequence using transfer RNA, which is tagged to particular amino acids, to build a linked sequence of them. One source of deep evidence for the common ancestry of all life on Earth is the fact that nearly all living things use the same genetic code to translate mRNA into polypeptides. There are rare, notable exceptions, but not so many as to make us think that the different lineages of taxa are derived from a different last common ancestor. Today we understand that phenotypic variation comes in large part from mutations in DNA, recombination, epigenetics, and developmental plasticity. Recall that mutations include many kinds of alterations to DNA, from simple and small-scale point mutations, or other kinds of mutations that can add or delete DNA content. Recombination happens during the development of the gametes of sexually reproducing species, but also happens through lateral or horizontal gene transfer in bacteria. Many species, including humans, also undergo somatic recombination of genes related to immunologic defense during development. Epigenetics is alterations to DNA expression that are caused by secondary modifications to the DNA. These alterations are heritable from one cell generation to the next, or even from one organismal generation to the next. Developmental plasticity includes the alterations to the phenotype that happen as a result of the local cellular conditions during growth, development, and remodeling. Rates of mutation vary from species to species, but also among tissue types, sexes, and between nuclear and mitochondrial or chloroplast DNA. In humans, the nuclear genome mutation rate is pretty rare, estimated to be from 1 to 3 mutations per 100 million nucleotides per generation. At one level, that seems like pretty high fidelity copying, but over a lifetime, that adds up to 60 to 80 mutations per individual per generation. On the other hand, not every mutation is a point mutation, and mutations in some parts of the DNA may be more significant than others. So the actual effects on the individual's phenotype and the genetics of a population are pretty hard to predict out of hand. From the time of Darwin on, much of the expansion of evolutionary theory developed because of the necessity to think in terms of populations rather than individuals. Even today, when a new species is named, part of the diagnosis and description of that species is based on reference individuals that are called types. However, it was exactly that kind of typological thinking that got the early natural philosophers into assuming that species had to be immutable. Today, we can make qualitative and quantitative assessments of change in biological populations. Whereas Darwin talked qualitatively about descent with modification, today we talk about evolution in living species in terms of changes in genotype frequencies in populations, and these happen over generational time. Notice here that we are seeing that individuals are born variable, and then selection is going to allow some of those individuals to survive to subsequent generations. However, on the right side of the panel, individuals do not evolve over their lifetimes. Individuals are born variable and then they are selected rather than changing during their lifetimes. The first step to describe how evolutionary change is happening. The first step to state that evolutionary change is happening is to be able to determine if it isn't taking place. What I mean by this is that it's necessary for us to have null models in science, expectations that we would have if there were no evolution taking place. Everyone gets taught the Hardy-Weinberg model in introductory biology and in genetics classes because it's a useful tool and a relatively simple mathematical algorithm for looking for deviations from expectations. This model looks for evidence of changes in a population's gene frequencies at a particular locus in a single generation or compared over generational time. This model should match the population's genotype frequencies unless one or more of these things is true about the population. The locus described is under selection, that there is non-random mating, there is mutation happening at that locus, there is gene flow, that is migration into or out of the population, or the population is small enough that the effects of random genetic drift are noticeable. So what you're seeing is these are all the interesting things, all the things that cause variation within populations. Just to give you a little bit of a baseline, this is a simulation of 5 different populations that are changing randomly through genetic drift. Notice that in population 2 and 3, this particular A1 allele becomes fixed, which means that it's present in 100% of the individuals in those populations. In 2 other populations, 1 and 5, that particular allele gets lost in those populations. In number 4, it continues on at about 60%. So it's possible for alleles to become present 100% of the time or 0% of the time in a population. Alleles can die out. And this kind of change can happen randomly or due to selection. We use the selection coefficient as a numerical descriptor to describe the intensity of selection. In these 3 panels here, I'm showing the effect of decreasing the fitness in the other allele, the A2 allele at this particular locus. So when you have a 10% reduction in the fitness of A2, notice that the frequency of the A1 allele starts at 50%, but then climbs pretty quickly over 150 or so generations to become pretty close to fixed. With a 40% reduction in the fitness of the other allele, that A1 allele hops right up within about 20 generations to become over 90% of the population. And if you decrease the reduction even further in the other allele, then the A1 allele swamps the population and becomes nearly fixed within about 10 generations. In addition to the effects of selection, we can also see how mutation rate can affect the allele frequencies. The top line of panels here is showing the frequency of that A1 allele when you have very low levels of mutation. So you see it mutating from A1 to A2. So notice the A1 starts at 50% and goes down to, about 45% of the population if you have very low rates of that turnover. With 10 times higher mutation, it scoops down pretty noticeably, and then with even 10 times more mutation, it's basically lost from the population within about 100 generations. The bottom panel is showing you for that same low, medium, and high level of mutations how the genotype frequency changes as well. Where we're going to go with this is to develop a conceptual model of what we call fitness landscapes or adaptive landscapes. Just like there are sometimes tortuous paths that we will take to go up a mountain, we see that gene frequencies will also go through similar kinds of switchbacks in order to get to higher fitness configurations.